I have just given a very brief summary of his research activities. He has almost like 16-20 pages CV. From that, I have extracted very small extract from his CV. That is, his current designation and affiliation is High Altitude Pulmonary and Pathology Institute, and also he is associated with Institute of Pulmonary Pathologia de la Altura, IPPA. And I know him in last year when we had jointly organized a world conference along with Dr. Malali Gowda of Bengaluru Genomic Center, and he was the resource person, and he had jointly organized the second COVID conference in last year. Apart from that, he has, like in 2016, he has organized and was a scientific member of 6 chronic hypoxia symposia organized in Bolivia. In 2018, he was the organizer and scientific committee member of 7th chronic hypoxia symposia at Bolivia. I personally thank Dr. Gustavo. Yesterday night, he agreed to deliver the talk today. I am heartily thankful to him. Professor Gustavo, you can start. I invite you to start the session. Thank you so much for accepting our invitation.
Thank you very much, Professor Bagchi. I am very honored. I actually came into your conference to learn about molecular docking, which definitely is the leading edge, one of the leading edge technologies that will promise a lot in the future. And what I have done here is prepare a talk to give you some background about my experience with high altitude and how it's related to COVID, as you have invited me. And so I'm going to give you a talk, and I will briefly talk a little bit about molecular docking and hypoxia. So, very good. I proceed to prepare. Oh, I need to, can you please make me a host so I can share my presentation?
Yes, sir. One second.
Okay.
I think you can now share your PPT.
Oh, now I have it. Yes.
Yes.
That's perfect. Okay. Let me see. Just a minute. Okay. Yes. Are you able to see my screen?
We can. Can you make it?
Okay. Perfect. So my talk is related to high altitude research applied to COVID-19. And as you can see, the light passes through because it has allowed us to see the light in many aspects of COVID, because we deal with hypoxia every day in our lives. By the way, next July the 9th, our institute will be 51 years old. So it's a very broad experience that I'm presenting to you now. If you see a patient that presents this type of pathologies in your lungs, you see how much this is supposed to all be sort of black, because the right side here, which is actually the left in the chest, is an area that's clear, but you see all the white spots there. And when you run a CAT scan, you can see this type of lesions in the lungs. And if you measure the oxygen, it's 72% saturation. This is in La Paz. The heart rate is 118. We measure D-dimer 2,906, C protein, reactive protein 6.8. What would be your diagnosis be? I'm pretty certain most would say COVID. Well, actually, 2 days later, the lungs are already cleared. You see all those images in the chests are clearing up. Here's the CAT scan. And 3 days later, the pulse oximetry with oxygen at 2 liters per minute is 99%. If we let the patient breathe on the air at high altitude, we have a 91%, which is absolutely normal in the city of La Paz. In fact, my saturation right now is 90% or 88 to 91 during the day, and that's our normal saturation. So this patient 3 days later has a normal condition at 3,500 meters. Diagnosis, high altitude pulmonary edema. You see, this is extremely interesting because it allows us to show we have been looking at these low oxygen levels in these high altitude pathologies from a long time ago. So we have extensive experience. And there is a very important thing. There is no pneumolysis. Do you see down here? There is no pneumolysis. That means that's what I'm going to talk about a little bit. This is our city of La Paz, located between 4,100 meters to 3,100 meters. You see many skyscrapers. You see the mountains around, many houses all around. This is during the night, and that's the beautiful Illimani that you see behind me, also in front of our city of La Paz, which is absolutely beautiful. These are the teleferics that it's like a metro system, but we go through the air. We have over 12 lines. We can move all around the city with these teleferics. And our saturation, like I told you, is typically between 88 and 92%. My daughter, who is also a medical doctor, has said that actually we live in silent hypoxemia, which was just discovered in COVID at sea level, but we have been seeing it all the time. So when you are at sea level, the oxygen levels are PaO2 is 98 millimeters of mercury, carbon dioxide is 40 millimeters of mercury, and your hematocrit can be around 42%. So what happens when you come to a city like La Paz, Bolivia, at the altitude of 3,500 meters or 3,600 meters, you arrive at the airport, we measure your oxygen levels, we do a blood gas study, and we measure that your oxygen is already at 60, or maybe a little higher, 65 if you're hyperventilating, which most are, of course. And carbon dioxide is 30. You see, it has decreased to the 10 millimeters of mercury. If you come up very fast, you can get acute mountain sickness, headaches, and so on. Or you can have the most serious high altitude pulmonary edema or high altitude cerebral edema that can be lethal if not treated properly. But in 51 years of practice, medical practice at our institute, we have never had a casualty or death from these diseases because we know how to handle it very well. And but if you come up to high altitude slowly, you take it easy a few days, your hematocrit in 40 days will rise to 50%. It takes a while for the red blood cells to be produced, so you adapt to high altitude. So you see, this is my normal hematocrit here in La Paz, 50%. And so we have a remarkable adaptation to chronic hypoxia. So what is the most important factor of lethality in COVID? Hypoxia. Why do people die in COVID? Hypoxia. Fundamentally, it can be fundamentally a lung disease, which is most of the cases, or it can be vascular lesions and obstructions, but it can affect many parts of the organism with the vascular lesions, the endothelial lesions. So what is the oxygen capture organ of the body? It's the lungs. Are there other organs involved? Yes, the oxygen transport triad, which we have described. The pneumodynamic pump are the lungs that are moving air from the environment into your lungs to transport oxygen inside and to blow out carbon dioxide. The hemodynamic pump captures the oxygen when it's going through the capillaries and transports the oxygen to all tissues, all the way up down to the mitochondria, through diffusion, of course. And hemoglobin is the fundamental factor. That's why we have called this the oxygen transport triad. The pneumodynamic pump, when it gets hit by the COVID virus, can produce the very severe pneumolysis. I will talk about that. This is a word I created to describe it. The hemodynamic pump can also be hit because the vessels can be hit, the nerves can be hit, and so myocarditis is another problem of the COVID. And of course, since it's the hemodynamic pump, it has to also do with hypoxia. And the vasculitis, that's a very important thing about inflammation in the vessels. So the protection factor becomes of this triad, the hemoglobin. Look, we have recently published a paper with some very interesting colleagues, Virue Soto, and from Peru, from different places, mostly South Americans, and with my daughter Natalia also, and where we say that low serum erythropoietin levels are associated with fatal COVID cases at 4,150 meters. You see here, these are the survivors in the white column and the deceased in the black column. Those that had higher erythropoietin survived much better. The hemoglobin went the same because it's related, as you know. Those with higher hemoglobin survived much more than those that had lower hemoglobin. Where is hypoxia best tolerated? Well, we wrote a very interesting paper called Tolerance to Hypoxia. You can look it up on the web. And it has to do that there's a high altitude paradox. Where is hypoxia best tolerated? Here at the beach, where the oxygen tension is 100, or in the summit of Mount Everest. At the beach, if your oxygen drops to 60, you're already in critical hypoxia, and you have to go into intensive care. But in La Paz, we live with 60 millimeters of mercury. These are normal oxygen pressure. And so on the summit of Mount Everest, oxygen tension is 24 millimeters mercury. And people have ascended to the summit of Mount Everest without oxygen, as you all know. Women also. So how is it possible that people can tolerate that? Well, we have the explanation for that. You see, at sea level, if you tolerate hypoxia once, on Mount Everest, you tolerate it 6 times more than at sea level. Why? Why? Well, it has to do with the tolerance to hypoxia formula, which we have created. It carries the numerator is the hemoglobin. In other words, if you have more hemoglobin, you're more tolerant to hypoxia. If you have a lower carbon dioxide, you are more tolerant to hypoxia. And so this is what we call the tolerance to hypoxia formula. Now, when I was a visiting professor at the University of Copenhagen, I spent the first 3 months, and then I came back to Bolivia to work for about a month and a half. And when I came back, my hematocrit was 36. Normally in La Paz, my hematocrit is 50. So you see, there is a process of adaptation, but it actually takes, for a full adaptation, it takes 40 days for me to recover my hematocrit, my normal red blood cell count, and hemoglobin, of course. It's a slow process. And during this stage, I can feel short of breath like sea level people when they come to high altitude. But once I achieve my normal hematocrit, I carry on with a normal, perfectly normal life. So the sea level inhabitant has a disability. He has poor tolerance to hypoxia because of the environmental conditions. You see, you have been adapted to live with a lot of oxygen. We have been adapted to live with little oxygen. So what happened? What is this disability? You have a higher carbon dioxide. I showed you in the formula, that's a factor. You have a lower hemoglobin. That's the other factor of tolerance of hypoxia. And you have plenty of oxygen. Whereas the high altitude inhabitant like us, by the way, La Paz is the marble city of the world, one of the 7 marvelous cities of the world. We have altitudes ranging from 4,100 to 3,100 and 2.7 million inhabitants, the highest population living at high altitude. The high altitude inhabitant, due to his permanent exposure to chronic hypoxia, has biologic mechanisms of survival that result favorable in multiple diseases, including COVID-19. In January the 31st, I was asked to participate in an interview, a radio interview regarding COVID. Initially, I rejected it because this was January 2020. We hardly knew anything about COVID. We only knew it was in Wuhan. But they called me again 5 minutes later and they said, you have to come. So I did. And this stimulated in me to dedicate all the time from then up to now to study COVID, to theorize, to give you our experience regarding hypoxia. In this experience, I said in this interview, I said that the high altitude inhabitant would have advantages because he's at high altitude, and also because we have a higher ultraviolet radiation. I wrote an article on April the 15th, 2020, saying that we would have advantages at high altitude in the control of COVID-19 due to high ultraviolet radiation. Why did I say that? Because when I was a visiting professor at the University of Copenhagen, we wrote an article with these colleagues, Danish colleagues, and this was presented where you can see in Copenhagen during the whole year, this is the amount of ultraviolet index radiation, and it's quite low. Whereas in La Paz, we have completely a different panorama. You see, in January, we have almost 18 ultraviolet index, and in the winter, our lowest is close to over 80 or 7. Sometimes it varies a little bit every year. But you see here, that's the great difference. And so the Condor of the Andes is a beautiful bird that flies at 6,500 meters of altitude. And he has this extremely accurate vision. And so with his vision, we have looked into the coronavirus and studied it from a different point of view of the highlands. We wrote an article which is published. It was a preprint on May the 5th. And by the way, this was published with my daughter Natalia Subieta, and also with Professor Tupil Venkatesh, the great lead man of India, who is a fantastic man that I have traveled with, and he has invited me to India 5 times already to give talks all around India, and Professor Kushal Das from the BLDE University, and Jorge Solis, who is a Bolivian now working as a professor at the University of Laval in Canada. So in this article entitled COVID-19 and Pneumolysis Simulating Extreme High Altitude Exposure with Altered Oxygen Transport Physiology, I talked about multiple diseases at the very beginning, scarce need of ventilators, and the Andean condor's eye view. I talked about multiple diseases because it doesn't only hit the lungs, it hits the vascular endothelium and that affects the nerves and it hits the brain. And so I talked about all these pathologies. I don't have time to give you all the details. And I talked about a lower incidence of COVID-19 at high altitude, we talked, and how COVID can float in the environment as it is an 120 nanometer organism, RNA molecule virus, and has spikes, the scarce need of ventilators, the oxygen transport triad, the post-COVID increase of hematocrit, hemoglobin, and red blood cells. After the lesions, a recovery procedure will be that hematocrit and hemoglobin has to go up to compensate for the lung deficiency and the important use of aspirin. Look what happened in Bolivia at the beginning of when it first arrived in March. The green line you see is the lowlands, 400 meters above sea level in Bolivia. You see how it went up so much. In La Paz, where we live, it was this line, that's this blue interrupted line. You see, we didn't have hardly any COVID at the beginning. Then it went up, but it didn't go up as it did in the lowlands. And this continues up to date. We wrote an article to the High Altitude Medicine and Biology journal showing that in Bolivia and Peru, the case fatality rate was 8.6 in the lowlands percent, and only 3.2 in the highlands. In Peru, it was close to 6 in the lowlands and 3 in the highlands. You see, we have lower case fatality rate. And so ultraviolet radiation C does not reach the surface of the Earth, but ultraviolet radiation B does, and it has a sterilizing germicidal effect efficiency of 70%. So it's real. You see, at the beginning of our pandemic here in La Paz, the levels of ultraviolet radiation at the time were very low. So that's why you saw that we didn't have much COVID. And as it gradually went down and down and down, and when it reached below 8, then we had many cases of COVID, as you have seen, but not as many as those in the lowlands anyway. And these are some information that I have obtained from this on the web. And you can see this, the ultraviolet index in October, and you see how high it is in this area in the south. Then in July, we are in the winter in the south area, but in the north and where India is located, that's when the ultraviolet radiation is higher. But if you look in the winter in the southern areas, particularly in the Andes, we have a very, very high index. It's the highest, above 18 ultraviolet radiation. And this has been one of the protective factors. You see, here is weather online of the city of La Paz in 2020. The lowest it went was above 6. We wrote an article with Cristian Arias Reyes and Jorge Solis, a very interesting paper, and my daughter Natalia and many Bolivians here, except Matthias Dutschmann, who's Australian. And in this article, Christian and Jorge proposed that also the ACTE2 receptors have been found in animal models, that they're at high altitude, there is a lower number of receptors. And so there was the probability that there was also a biologic effect of protection at high altitude, which was very interesting, of course. This is one of the most cited papers in relation to high altitude and COVID now around the world. It has been cited very, very often. And there's another article that Cristian Arias with Jorge Solis developed, and which we had the honor of participating here also, and which says that there's a decreased incidence, virus transmission, and severity of COVID at altitude in the American continent. By the way, that was a preprint, but that's already has been published, and it's printed in a very good journal. So what happens is that as you go higher, there is less COVID. You see that? That's very interesting. Here, about 1,000, much lower incidence of COVID cases. And look at this that we have looked into with the physicist Roberto Vera from Potosí, from the university in Potosí, where we see here the ultraviolet index in black and in red the number of cases. You see, when on January the 7th, we had a drop of the ultraviolet index, and then 2 weeks later, you see, we had a rise in the number of cases of COVID in Potosí. Potosí is at 4,100 meters above sea level. It's one of the cities in Bolivia. And what has happened also when the ultraviolet index went up? 2 weeks later, we had a drop. So you see, we are finding a correlation between ultraviolet indexes. And so I checked in 128 countries all over the world, I averaged the ultraviolet index per country and plotted it about the case against the cases per population. And this is what I found in 128 countries, that we would decrease as the ultraviolet radiation became higher, we would decrease in the incidence of COVID. It's a fact for me, but it's one of the variables. Now look, ultraviolet radiation has a photoelectric effect in metals that the brilliant Einstein demonstrated. Blue light was able to have a photoelectric effect, and that's because it's approaching ultraviolet radiation, you see? And I have named this the photoelectric biologic effect on life, which is well known, but acts on plants, producing photosynthesis and the wonderful oxygen production from carbon dioxide. And this photoelectric effect also acts on animals and humans, and bacteria and viruses, of course, in the animals and humans producing vitamin D, extremely important. I treat my patients, COVID patients, and tell them, you must expose yourself 10 to 15 minutes, a larger area of your skin to direct sunlight every day, because we need vitamin D. That's why we have stronger bones at high altitude. And also it's an immunity boost. And as you know, immunity protects us against the viruses. So at high altitude, what happens with this? We have a higher immunity boost because the photoelectric biologic effect at high altitude is much higher, affects bacteria and viruses floating around in the environment outdoors. And of course, we have a greater immunity boost, which is fantastic. So confirmed, at high altitude, we have a lower incidence and case fatality rate of COVID-19. This is a paper that we're about to publish. We have studied the populations in Bolivia, Colombia, Ecuador, Peru, and Mexico. 161 million people were studied to study what the incidence was and the case fatality rate of all these highlands and lowlands, separating this is part of the study, a multinational study with all the people participating. And what we found, this is just a very small area because it's going to be published very soon. Lethality you see in Bolivia, you see the pipes here lower than 1,000 meters and higher than 2,500 meters. In all of them, you see that it's much lower in the highlands that we have lethality, lower lethality. Now at sea level, an SpO2 of less than 90 and shortness of breath puts you into a ventilator. In high altitude, we live with a 90% saturation, and so we are normally with these pressures of oxygen. Now let me talk very, very briefly about pneumolysis. Pneumolysis is the effect that I have described of the virus on the lungs, because it actually produces, this virus produces lung destruction. It's an extreme viral aggression that actually destroys the alveoli because it goes in the alveoli, as I will show you, and it's very severe. And if we destroy the alveoli, you reduce the surface area of the lungs of gas exchange. Normally, if we extend all our lungs we have like a tennis court, the surface area, so that oxygen enters into our body, because we have alveolar sacs. But the COVID attacks the surface area and starts reducing the area. So it is as though the highest point of extreme tolerable hypoxia where life is possible is at the summit of Mount Everest. But if you're at sea level and you get attacked by the COVID virus, it's as though you were put on the summit of Mount Everest, but with not enough hemoglobin and excessive carbon dioxide, the two tolerance to hypoxia factors. And so you go into critical hypoxia, and obviously that can result in the end of life. We're very sorry for all the people that have lost their lives, and it was due to hypoxia, low oxygen. Now look at high altitude pulmonary edema. In this case, that he was an European guy, that he was a rugby player and came and had high altitude pulmonary edema. When we measured it, his saturation was 54, his pulse rate was 107. Actually, it went down to 45. I also have another photo about it. And he was walking around and telling us he was not feeling well, coughing a bit, and so on. So his oxygen levels were 34 and his saturation 53. But next day, with beginning of treatment, oxygen and so on, his saturation was already rising at 76, breathing ambient air, which was quite significant change, as you can see. And you see, these patients come with their lungs like this, with an oxygen level very low, and in 48 hours, they can have their lungs completely cleared. So let me tell you, in COVID, it's a completely different story. You see the lungs get hit here very severely by the virus, and it spreads all around, and the surface area is obviously severely compromised. Now, let me show you with this how the virus goes into the alveoli, into the lungs, and it goes into this beautiful membrane that is very specialized. This is the alveoli, and these are the capillaries, and oxygen has to go through from this air to the blood here to be captured by hemoglobin. So there's ACE2 receptors. This is at sea level, but in the highlands, theoretically, we have less ACE2 receptors, and it's one of the factors why we have less problems. And so these viruses go in the cells to reproduce themselves. They change the program, the software of the cells, and they start reproducing within our lung cells, you see. And then they break the cells and they come out and spread throughout the rest of the lung. This is why they spread so fast. And this produces a vasodilation, but it also produces, with this pneumolysis, destruction of the cells, an entering of this virus to the blood vessels, and it also hits the ACE2 receptors, and that's why it produces vasculitis and thrombotic problems also in different areas. Now, very briefly, I will explain to you that oxygen goes down. This is the pathophysiology that we have developed with my daughter, Natalia. You see, these are blood gas measurements. Anyway, oxygen goes down, down, down as the lungs get more and more and more compromised. And you see this drop of oxygen is at the beginning well tolerated. This is why they talk about silent hypoxemia. But it gets to a point where carbon dioxide starts rising and it becomes very severe. And of course, breathing becomes extremely severe, and you can go into the death zone where you lose your life because your lungs have been so compromised, you don't get enough oxygen. At high altitude, this was the sea level curve, remember? At high altitude, we already start with a lower oxygen. So we have a better defense against low oxygen levels. That's why we publish these papers. By the way, this is in the Indian Journal of Clinical Biochemistry. And so it becomes evident that oxygen, the simple molecule that's called oxygen that's all around us, becomes more valuable than gold for life. And that's a teaching of nature. This is our adaptation formula to high altitude. It's time over altitude. In other words, if you have more time to adapt, it's better. The more altitude you go, the more time you need to adapt. Now, we wrote an article called Acute Mountain Sickness, High Altitude Pulmonary Edema, High Altitude Cerebral Edema View from the High Andes to teach the world how we treat patients. And over 51 years, successfully, I have recently treated a patient from an embassy that had a pulmonary edema, and it was following my guidelines. In 2 days, in 3 days, the recovery was great. But what has happened with this is that sea level high altitude experts have been upset because they think they see hypoxia differently from sea level in relation to high altitude, you see? They don't understand hypoxia the way we understand it and the way it has helped us in COVID understand it. So they wrote to the chief editor of this journal that this article should be retracted because they don't understand it. So you see, these experts from French, Americans, Chileans, Peruvian French, Peruvian English, one Spanish, Italian, one Nepalese, one Chinese. Well, the Nepalese, for instance, he lives in Kathmandu, and it's at 1,300 meters, if I'm not mistaken. For us, that is lowlands, you see. According to Peter Barr's classification, 76% of all these people live at sea level. 2 are from low altitude, that's 1,700. 1 is from moderate altitude, 2,200 meters. I think Kathmandu is 1,700 meters. And 1 from high altitude, but he has no papers in this area. But just to give you an example, we described that in high altitude pulmonary edema, the tongue has these lesions like that. And we have called it the Hape tongue, and we have associated it to a viral aggression also. Now look at the COVID tongue. Nowadays, the COVID tongue Look how similar that is to that. So there's a similarity of these diseases, although many of these sea-level experts have published that there is absolutely no reference, it's not the same pathology, but it is. The hypoxia is the same. Hypoxia is the same either in high-altitude pulmonary edema or COVID disease, but with different consequences because of the pneumolysis. This is my father working in isolated hearts of dogs in 1964, showing that the dogs of hearts are much more resistant at high altitude than at sea level. And this is true now. We have lower incidence of myocardial infarction at high altitude. This is our pyramid laboratory at 2,665 meters. It's the highest laboratory in the world, and we do studies there. Now, very briefly, since Dr. Bakci invited me to talk in your conference, I want to touch very briefly. I don't have much experience in these areas, but talking about the genetic adaptation to high altitude, there's a natural selection in several gene regions influencing the oxygen-sensitive pathways, and it's been looked into by many people working with genetics. You see, there's a Tibetan model and an Andean model. There are differences. There's lower hemoglobin in the Tibetans, apparently, lower pulmonary pressure, and less chronic mountain sickness. In the Andean model where we are, we have higher hemoglobin levels, higher pulmonary artery pressures, more chronic mountain sickness, apparently. But although my father, when we went we were invited to a conference, the World Conference in Lhasa, Tibet. As we got out of the plane, we saw people with chronic mountain sickness that were playing the drums, and I have a photo of that. Regardless of the mechanisms, both do survive at high altitude with different genetic traits, but we adapt. We all adapt to high altitude. Now, molecular docking at high altitude. You see, in one of my papers, we have proposed that the use of the term high altitude, HGE, high altitude gas expansion. This is very important because when the barometric pressure decreases at high altitude, gases expand. You see, in the city of La Paz at 3,600 meters, according to the Boyle's law of gases, due to the lower barometric pressure, maintaining the same body temperature, if we use this Boyle's law, which is the pressure times the volume, volume one at one condition, and then if we change the pressure to, we want to see the volume 2. And so if we calculate this, we put the values of 760 millimeters at sea level, and we multiply by a liter of gas. If we inflate a balloon, a liter-sized balloon at sea level, and we bring it to the city of La Paz, it will become 1.5 times bigger. In other words, it will grow 50%. You see how the gases expand here? And now, molecules are more distant from each other because of a lower barometric pressure. So in relation to molecular docking at high altitude, I have given it some thought. In air, molecules expand 50% of the volume as compared to sea level. Like I told you, at sea level, 1 liter of gas becomes 1.5 liters of gas in La Paz. But in water, in fluid, in liquids, it's different. At sea level, 10 meters if you dive 10 meters at sea level, you increase 1 atmosphere of the pressure. So that would be 2 atmospheres actually, but it's 1 atmosphere of pressure in 10 meters of water. If we dive here in the city of La Paz, we have Lake Titicaca, where the highest navigable lake at 3,800 meters. In fact, I have with Professor Paul-Erik Paule, we have developed high altitude diving tables because the decompression sickness can be much more severe. If you dive 40 meters in Lake Titicaca, it's like diving 70 meters at sea level, the risks. So you see, in La Paz or in Titicaca Lake, if you dive 3 meters, you are at the pressure of sea level. So the pressure in fluids is different. However, you see, if you look at molecular docking at high altitude, at sea level, if you have this molecule, a molecule like this, is it probable that there will be a variable at high altitude where the molecules will have to accommodate differently because of a lower pressure? This is a question for you. You are the experts. And and I have looked, thanks to Professor Venkatesh, whom I commented that I had been invited to be in your talk, which honors me greatly. And I'm almost finished here with my talk now. I looked at, and he mentioned that we talked a little bit about this, we communicated, and he gave me a clue that molecular docking in pharmacophore models to pro-binding hypothesis of inhibitors of hypoxia-inducible factor. You see, this is another one, use of network pharmacology and molecular docking to investigate the mechanism by which ginseng ameliorates hypoxia. So hypoxia is being the focus of molecular docking, and it will become the main focus now, even after this COVID. So you are in a wonderful field of leading edge technology, and I'm very honored to be joining you because I'm learning very much from you. These are the altitude laboratories of the world, and our Chacaltaya pyramid is the highest in the world currently. We are studying here the high altitude divers. We have the highest altitude diving team, the school from the Bolivian Navy. And you see, we are studying them in our laboratory, performing blood gases to measure the oxygen levels. And I have we have published several papers. One of these is, do over 200 million healthy people, residents, really suffer from chronic acid-base disorders? This was with Paul-Erik Pauleb, and then we're here with Ole Sigurd Andersen, the man that developed many of the aspects of acid-base balance in Copenhagen. This is at the University of Copenhagen, very beautiful memories. And you see here how we at high altitude live longer than the people from the lowlands. This is very interesting because this is Bolivia, and I'm showing here increasing altitudes according to different cities. And you can see comparing two cities in the lowlands at 416 with similar populations to La Paz. You see we have, these are the older than 90 aged people. You see how many more we have, and these are the older than 100. So we have written a paper at the BLDE University Journal in India, where I'm a visiting professor, thanks to Professor Kursal Das and Professor Biradar, who unfortunately left us recently due to COVID. We participated in a memorial for him that extended longevity of high altitude and many benefits of chronic hypoxia. By the way, this was during the Abicon 2017 opening ceremony, clinical biochemistry, and this is Professor Vice Chancellor Bat, King George University, Professor Ali, and so on, and we were lighting the candle, which I love. Incidentally, I'm using here the quote that Professor Venkatesh lent me because I hadn't taken a quote to this conference. And at that event, Professor Bat, when I was at the university, named me the hypoxia man because it was easier probably than remembering my name. Yes. And here is my father, which I honor always, because what you see here in my talk is his education, what he has taught me. He passed away in 2015. I want to thank Natalia Subieta de Ros, my daughter, my wife, Lucrecia de Roste, my other daughter, Lucia, and they are main collaborators, and of course, many other students that I have. And I always ask, because we don't have any support, financial support here in Bolivia for science. We do everything out of passion from our own work. Over 51 years, we have never received a grant because we don't have access to them. So we would appreciate any support you can give us. Thank you very much, and I hope I am on the perfect timing here.
Thank you so much, Professor. It was really very, very interesting talk. So let me ask the participants if you have any questions to the speaker. You can I can ask the question, or you can even put it in the chat box. I'll read the questions.
Okay. not the type of talk you would have in a molecular docking conference generally.
Yeah, it was a special talk.
But Professor Bakci, I find it interesting that there are some they're beginning to look at molecular docking with hypoxia. I think that's fascinating.
Yeah, I am waiting for any question.
Right. Oh, I'm happy I'm reading some of the chat messages, and yes, I'm happy that I was able to communicate with you. Thanks to Dr. Bakchi that invited me. Thank you so much. It was great to give you a little bit more of what we have been looking at recently. Yes, and many of you have been in my previous talk, so it's wonderful. So, Professor Bakchi, it's very interesting that at high altitude, we need to look more at molecular docking, but in a different environment, which is very, very, I think it's a new field.
Yeah, definitely we have to study that, because I never, this is the first time I seen that lines, molecular docking in high altitude. So let me
Yeah,
explore that, and very soon we will be collaborating with you in this regard. Because
very
actually we
interesting.
don't have any data on in this area, that is on high altitude, but definitely we will be looking forward to this. Okay, finally we have a question. Sir, after becoming COVID positive, whether going to high altitude will help or not?
Well, that's an important question. I would not advise anybody that has a serious acute COVID disease to go to high altitude, because you would aggravate your hypoxia, of course. But let me tell you that after you pass, if you survive COVID at sea level, you may remain with some lesions in your lung with fibrosis, pulmonary fibrosis. And if you have this fibrosis, you will have some shortness of breath. So you need more hemoglobin. This is very important. I didn't have time to talk about it. We are, you see, coming to high altitude gradually would be a wonderful stimulus for your body for recovery post-COVID, because it will stimulate ventilation, it will stimulate an increase of erythropoietin, and will stimulate a better oxygen transport, which will be beneficial. Now, this is of course in the preliminary sessions, but it's part of the physiotherapy of respiration, breathing again. And I am wondering if the lungs that have been destroyed by COVID, if they have fibrosis, they cannot recover because it's a scar in the lungs. But certain areas that have had inflammation are able to recover. So it's very interesting.
Yes, thank you, Professor. There is one more question. How do these high altitude people get adapted?
I'm sorry?
How do these high altitude people get adapted?
How do we adapt is the question, right?
Adapt, yes.
Okay, our adaptation has to do with in the acute phase, the pneumodynamic pump, which is the lungs. You hyperventilate more, you breathe more. When you arrive to a high altitude area, you breathe more. Your heart pumps more. This is in order to achieve a better capture of oxygen molecules, because there's a lower pressure. But gradually, with time, the red blood cells begin to increase gradually, step by step, and once they increase to your optimal level, which is the maximum, more efficient level for your increase of red blood cells, then you adapt very well to high altitude. Let me tell you, my grandmother had a father who was from Spain. He came to Bolivia to live, and he remained forever. And he had 3 daughters. The oldest lived to be 100. The other one 99, and another one 98 at high altitude in the city of La Paz. So people that come from sea level can adapt quite well also, except when there is some kind of disease that has to be diagnosed, of course. Good question. I see there's one more, Dr. Bakchi.
Yes, Dr. Rashmi has asked a question.
What is the optimum hemoglobin count to avoid the COVID infection? Ah, that's interesting. Well, you can't avoid it, even though you may have high. But the higher your hemoglobin is, the better your chance of survival. That's what we showed in that paper I presented at the beginning that we recently published. You see, the higher hemoglobin you have, you have more oxygen reserve pool. That means you have more red blood cells. And if you give oxygen to the person that is suffering COVID and this person has low hemoglobin, this person cannot capture a great amount of oxygen to transport to tissues. But if she has a higher hemoglobin, they can capture much more oxygen. This is a beautiful thing, and I have named it the oxygen reserve pool. In other words, the more hemoglobin you have, the more oxygen your cells can transport, and that can save you while your immune system attacks the virus, stops the lesions in the lungs, in the vascular endothelium and so on. And that in the meantime, your system starts recovering. So higher hemoglobins are important. My normal hemoglobin is 16 grams per cent here in La Paz. So as you can see, that's quite high for sea-level people, but the good nutrition based on the good nutrients is really important. Oh, there's another question. How different receptors for spike proteins express in high altitude? Wow, that's a tough question. I don't have truly a very precise response to that. I think it's an important area of research, Dr. Mohanty. Ah, that's another interesting question. I don't know if we still have time, Professor Bakchi.
Yeah, yes, please continue.
Curious to know why there is permanent binding of hemoglobin with carbon monoxide rather than oxygen carbon dioxide. Ah, that's a good question. I like that. The thing is that carbon monoxide gets very strongly attracted to hemoglobin. And why does this come? It has to do with the characteristics of the oxygen capture with iron, how iron links to oxygen, the electrons and so on. It's a little complicated to give you a very detailed talk about this. But let me mention a very interesting thing, and I love the idea you asked me this now. Yesterday, I was looking into a paper of some forensic pathologists that had studied some people that had high altitude pulmonary edema in the mountain. And it happens that they found a man that had died apparently from high altitude pulmonary edema in his tent. And he had his 3 children with him, I think. He and one of the child also died and 2 survived. But they found in the tent, they had put in a gas stove to warm up because they were very cold. And they went to sleep with the heating stove. So what happened? They actually did not die from only pulmonary edema because they did have some pulmonary edema, which is logical, but they died from carbon monoxide poisoning. You see, carbon monoxide links to the hemoglobin, and it produces a very bright red color of the blood. It's the characteristics of the colors. You see the molecular addition of oxygen, of carbon monoxide to hemoglobin produces this very, very particular, highly reddish color. But that blocks away the possibility of transport of oxygen, and it also gets very strongly adhered, so it doesn't come out very easily. So this man did not die from high altitude pulmonary edema, primarily. He died from carbon monoxide poisoning. So obviously, you should never use any type of heating based on gas furnaces that produce carbon monoxide indoors in an enclosed environment. Very, very dangerous. Yes, your other question is extremely good. If you stimulate hemoglobin synthesis by any means, you provide better protection for COVID patients. Let me tell you that we have been using here erythropoietin. Erythropoietin is a hormone that we all have in our bodies that is generated in many cells in the body. Originally, it was found that it was generated only in the kidney, but now we know it's all around. Professor Solis is an expert in erythropoietin at the University of Laval. And you see, I have been using erythropoietin in patients that have low hemoglobins or poor response to hemoglobin, and that improves their quality of life even in pulmonary fibrosis patients. I have been using it for several years now. And in this COVID pandemic, I give my patients also erythropoietin, and it has given me great results. So the idea is to improve the red blood cell count in COVID, erythropoietin, but it has to be handled by physicians always, because you have to look into other problems that could arise. But it's generally very well accepted. I have never had any problems with it. And I have had success with all the patients I'm treating by using also erythropoietin. I don't know about moringa soup. What is the effect? What is moringa soup? And maybe it's a very interesting thing. Can you expand on moringa soup?
Can you please unmute and speak?
Yes, KV. KV, can you speak? Please, Dr. KV? We cannot hear you. Unmute yourself, please.
So
Oh, it is said to...
yeah,
Yes.
he's saying moringa has a high hemoglobin content.
It has hemoglobin, or you mean it really? I don't know about it. Is it from meat, Dr. KB?
It's a plant actually, sir. It's a drumstick. As a vegetable, it is known as drumstick. That's what he's mentioning. That
You
plant.
mean if you have the plant, if you have the soup, it stimulates red blood cell production? Is that what you're saying?
Yes, sir. The leaves, they make a decoction of that, or a soup kind of a thing. They eat. In Indian food, they use these leaves, drumstick leaves.
How interesting. You know, well, first of all, I love Indian food. I'm in love with Indian food. Professor Venkatesh has introduced me to the most delicious food all over India, which is absolutely great. So I would like to know a little bit more about this moringa soup. It sounds very interesting. If it does stimulate hemoglobin production, would be extremely useful in COVID.
I can just show you the picture. I will just share the screen in a minute. You can see the picture.
Okay, let's see it. Okay. Oh, how interesting.
We call it
That's
drumstick.
moringa.
Yeah.
Yes,
Yeah,
yes.
this one also can have a better look.
Actually, in
Yes.
Africa, they grow it as a crop, and they grow it like any other crop.
How nice. Very interesting. Very interesting. Do you know of any studies that actually it increases the hemoglobin?
Actually, moringa leaves are rich in iron content, and it is usually used to treat anemia in India.
Ah, okay, okay. Yes. In order for hemoglobin to increase, you absolutely need good iron, but you also need B12. B12 is because the hemoglobin molecule requires B12, vitamin B12.
Yes, it is also rich in vitamins and calcium as well.
Calcium also. Yes, very interesting. Thank you very much, Dr. KB. Very interesting.
Okay, if there are no further questions, then I would like to thank Dr. Gustavo on behalf of all. And we will be starting the next session in 1 or 2 minutes, that is the sequence alignment.
Thank you, Dr. Vakci. It was wonderful to speak with you and to share some moments with all your distinguished assistants to this conference.
Yes, thank you so much. Thank you so much, Doctor. It was really interesting. Yes, please, somebody has raised the hand. You can unmute and speak. Somo Buddha Roy, you want to speak something? You can write it, put it in the chat box.
Actually, there is one question. What will be the effect of high altitude on skeletal muscle energy metabolism?
Oh, yeah, that's an interesting one also because you see, we have at high altitude less obesity because the organism has to adapt to an efficient energy use of oxygen. We carried out a soccer game on the summit of Mount Sahama at 6,542 meters. The guys that played were the porters that carry always in the mountain the oxygen tanks and the loads that the people that climb up require. And they, if you look at them, they are all basically very little fat in their bodies and mostly muscles. So the adaptation of the skeletal muscle is also very peculiar at high altitude and very favorable because we are much healthier in many aspects. And I'm happy you point out that question, Dr. Verma, because we have many advantages of health at high altitude. I have mentioned to you that because we have more vascularity, you see, we have the endothelial vascular endothelial growth factor is a stimulation, just like hypoxia-inducible factor is a stimulation that arises from, it's an epigenetic response to hypoxia, which allows the physiologic adaptation. The increase of vascularity at high altitude is a very favorable condition, and it's in all tissues, even the brain, and also in the heart. You see, the people at high altitude have more vascularity, and so we have a lower incidence of myocardial infarction at high altitude, for instance. So muscles also receive more oxygen by more hemoglobin and more vascularity. Those are two fundamental factors of compensation. Okay, Dr. KB mentions also coconut milk and cloves. That's interesting. I'm sure that some nutrients are fundamental. I think that nutrients can have two effects with respect to COVID. One would be a direct effect on the virus. Some may be varicidal. In other words, they would destroy viruses. They would also have an effect, perhaps, of blocking the entrance of viruses to the lungs or other cells in the body. In other words, interfering with the ACE2 receptors. By the way, I have recently read an article in which I agree, and that is because we have been talking about this with Dr. Murray from the University of Miami, who's an expert in hypertension. You see, the use of hypotensive drugs, because they do link to the ACE2 receptors, can help block the entrance of viruses. And lately, I have begun to use in my patients some hypotensive drugs, because that way you would avoid that the viruses spread in the lungs. And it's very interesting. There's a paper from some Argentinian colleagues that have studied this, and they have found that evidently those patients treated with hypotensive drugs like losartan, telmisartan, they have had lower complications and better recovery. So I'm using now hypotensive drugs regularly because this does not affect the organism of the people, but it can help block the entrance. And the other factor perhaps has to do with an immune stimulation of food in COVID, which is extremely important. The immunity is our best tool. That's why vaccines are created, to induce immunity. But we also have natural immunity. And our natural immunity has made beings survive even the Spanish flu, which killed millions, but many millions survived. Why did those many millions survive? Because they had strong immune systems and they had the time to develop their immunity. The time factor is crucial. Yes, absolutely. We need more experimental evidence of that, some things, yes. Okay, I think we're done, Dr. Bakchi.
Yes, Professor. So finally, I thank you for this wonderful talk and patiently addressing to all the queries which you are getting. So we will see you again very soon for any other future events.
Yes, yeah, happy to join you, Dr. Bakchi.
Yes, thank you.
Thank you very much.
Professor. Yes. So we will start with the next session, that is sequence alignment. Give me a moment, please. Okay, good evening everybody. I am starting the sequence alignment. Now, I don't have any slides particular, but I will be explaining directly from the PPT, from the websites and the literatures. So, what is a sequence alignment? It is like alignment of one sequence with respect to other. So, we have to take one sequence as the reference sequence and we have to align the other sequence with respect to the first sequence. That is, if at all there are two sequences like sequence 1 and sequence 2, we can align the sequence 2 with respect to sequence 1. So that is how the alignment is. That is how the alignment is done. It is the alignment is same for the faster sequence as well as for the fast Q sequence. Even with the fast Q, there is a step called very important step called mapping. Mapping means nothing but it is the same like it is the alignment step. So in the okay. So what I have done is I have put the sequence 1. I will just make it little bit yeah. So I have put the sequence 1 as it is. I have given little spaces and then I have put the sequence 2 just below the sequence 1. So you can see here sequence A, it is as it is. So you can see here T, I have put it under T because if the T is matching with T, then I have G here. G I have placed under G. I can place it anywhere, but I have to put under G here. When you are using the software, it takes a specific algorithm and then it will assign in any of the G's which is favorable for the particular algorithm. In the same manner, I have put here as T and so on. So this is the alignment. I have aligned the sequence 2 with respect to sequence 1. Now what I have done is I have put here as dash. These dashes are nothing, these are the gaps. So once the sequence is moved here in the alignment, so once the sequence is moved here, a gap is created. So this lines are representing the gaps here, and these are all representing the perfect match. So these are all the perfect matches, whichever is has aligned. And here is something that I have put G below A. So since G and A are in the sharing almost the same function, that is both are purines, so it will form something called a semi-match. In sequence alignment, there are three matches happening. One is perfect match. So perfect match, usually the softwares they give us the star sign. Star means perfect match. Then you have semi-match. Usually the software give us color. It depends on the software to software. So semi-match, it is called as functional identity or similarity. Perfect match is a structural as well. And then we have light match. Light match, it comes, I will not take the light match here because light match tomorrow I will be taking the global alignment, multiple sequence alignment. There I will be taking the light match. It usually comes there. And next we have this gaps. Gaps are, it is represented by this gap, there is absence of sequence. And there is one more term called mismatch. Mismatch also, it is difficult, it may not always come in the pairwise alignment. Still, mismatch, there will be the sequences, there will be mismatch will be taking place. So usually these are the four, and one more, as I told, there will be one more type, that is light match. So in the sequence alignment, usually we have to look forward the perfect match, semi-match, gaps, mismatch, and light match. So when you are aligning two or more, usually for the pairwise alignment, we are aligning two sequences. So for the two sequences, so whenever there is matches are created, you will be finding it based on the symbols. So let us see the using the softwares. So I have taken two sequences of hemoglobin. So this is one sequence. So I am using this pairwise alignment. Pairwise alignment, as the name suggests, it is for two sequence. I did not open here. I will show you here in a better manner. So pairwise, you can see the definition which is given here. Pairwise alignment is used to identify regions of similarity that may indicate functional, structural, or evolutionary relationship between two biological sequences. That is either protein or nucleotide. And there is one more called multiple sequence alignment that I will be taking tomorrow. It is alignment of 3 or more biological sequences of similar length. So that multiple sequence alignment I will take tomorrow in the next class. So for the global, we have the needle. It is called it follows the Needleman-Wunsch algorithm. And there is one more. It is a nearly law, that is stretcher. It uses a modification of the Needleman-Wunsch algorithm. And again, we have this local alignment. It uses Smith-Waterman algorithm, that is water. And then we have matcher. There is no particular algorithm. It is rigorous algorithm, and it is built on LLN. LLN is again one more software for alignment. So LLN, I will not be using it. I will be using this needle as well as water. So I will show you. So I have taken the protein sequence here. So it can be both the protein and the DNA sequence. So I am pasting the sequence. So what I will make a little modification in the sequence. So there is one sequence and one more sequence I have to paste here. So I am removing the complete information, only I am just keeping the name of the organism. So it is just that software will be taking, because if it is the name is more, the output will be a little bit difficult to understand. So on same sequence I am copying in the water. So I will show you the difference, how it is happening. So I will be running it. If there is any problem, it will be shown. Yes, for the needle we have got the alignment. So this was the global alignment which we have taken. So you can see here for the pair, for the perfect match, there is no symbol as such. It is given that one single line is here. That means that these are the perfect. I will just make it a little bit larger. Okay. So these are the gaps as I showed you. These are the gaps, and this is the perfect match. This is the mismatch here actually, and this is the semi-match. So semi-match is usually represented by the colons, and these are all the gaps here. And how by seeing the alignment, I can understand that this is a global alignment. Why? Because the sequence is starting directly from one. Global and local means in the global there will be complete match for end-to-end matches. Alignment will be there. Whereas in local there will be the alignment will be there only in the particular region where the match will be there. So since it is a global alignment, you can see that the alignment is starting from the first sequence and it is ending in the last sequence. So that is why it is a global alignment. So coming to the local, you can see that only the region where the alignment is there, it is displayed. So it is, you can see that it is the first one, it is starting at sequence number 42, and second one is starting from sequence number 27. The aligned regions are almost the same. That is, this is the perfect match here. These are the mismatches. Here you will get the semi-match and so on. So coming to the legends here, the sequence length is, the alignment range is 369. Identities are 7.3, that is 28 out of 369%. They are identical. Identical, similar means, identity means there will be structural, functional identity. Similarity means there will be only functional identity, but there will not be any structural identity, and gaps. So how many gaps are there? So out of 369 sequence alignment, there are 281 gaps. So here only in the local alignment, only the, as I told, only the region has matched. So it is the 88 sequence are only matched here. In that there are 24 identical sequences and you have 37. So 37 are the similarity sequence. Similarity, as I told, these are the similar sequences. These are the similar sequences. These are all the identical sequence. This is the identical and these are the similar. And as you know, these are the gaps. Okay, so this is, I am ending the pairwise as well as the pairwise alignment, that is the local and the global. So if you want to ask any questions, you can ask me. So tomorrow I will be taking the global sequence alignment, sorry, multiple sequence alignment and the phylogeny. So any questions for the class, you can ask me. Okay, there are no questions for the class. Okay, then I think tomorrow.
I would like to ask a question, Dr. Bakshi.
Yes, please, Dr.
Very interesting. As I told you, I thank you very much for teaching us all these things, because this is very advanced know-how. And I find that the papers published in relation to high altitude and the search for genetic patterns, you know, they're looking for genes of adaptation. And I find it's like a labyrinth, so many variables. They find different things and everybody concludes, yes, this is it. But then there are many more. And so it's an extremely complex science. Is this the habitual what's going on? Because like I tell you, it's a labyrinth in high altitude medicine to find the adaptation genes.
I could not understand the question. What asked?
Okay, so what I'm saying is that many scientists are doing genetic studies of people living at high altitude in Tibet and in the Andes, and they compare it to sea level genes. Okay. And so they try to find variables that are significant that would explain the advantages of life at high altitude. I mean, the genetic traits that would make people living at high altitude at an advantage genetic trait. So, but what happens, Dr. Bakshi, is that there is too many variables, and nothing is absolutely certain, because there are probabilities all the time. And so how much can do you to be effective in affirming that a gene has a significant value? How much similarity do you think you have to find or differences? Is that a good question?
See, if at all you are comparing the genes from the higher altitude and the genes which are present in the lower altitude,
Yes,
similarity I will not suggest. I will directly ask for the where are the variations. So
right.
based on whether the variations are there, we will look for directly the SLPs. So variants, if you find out, then we can come to a conclusion. Okay, these are the variants which are actually making the genes at higher altitude, and these may be more effective for recepting the oxygens. So in this manner, we can take, rather than coming to a similarity, we can come to the variation, and then we can come to a similar, in the other way around, can come to a similarity, saying that okay, this is to what extent the variants which are present in the genes in the higher altitude have similarity with the variants of the genes which are present in the lower altitude. In this manner, we can make a comparison based on the variants which are present in the genes, rather than directly going with the similarity which are present in the genes. So we will. It is always better to pinpoint and look in the variants.
How interesting.
This is what I feel.
Very interesting. Thank you very
Yeah,
much, Dr.
yeah.
Bhatt.
Thank you. There is one maybe one feedback. Are the beta and alpha chains of the hemoglobin molecule homologous, paralogous, or orthologous to each other? Actually, I have taken a random sequence from the NCBI. Uh, but you have asked the homologous. I have to check it out, frankly, because I just took the, because for the alignment I took the two sequences. Then you have to look it out how if they are homologous or not, because they are homologous in much manner, but to look for the others, because I just took the sequences without seeing, just for the alignment. In local alignment, on which basis the length? I did not choose the length on any basis. I just had to show you the difference between the local and the global. That is why I took one short sequence and one long sequence, so that you can understand the difference between the local and the global. Okay. Any other questions? Any other questions? This actually orthologous, homologous, and paralogous will not make come much in the sequence alignment study, but definitely it will have much effect when you are modeling the 3D structure. So in the sequence alignment, only the specific batches will be shown. If there is this one homologous, paralogous, orthologous, it will directly show, it come in the picture when it is like homology modeling will be taking place using the sequences. And if at all we have to study the more similarity between the genes, in that case also you can look directly for the homologous sequences. Okay, did I answer to both of your questions, Redna and Shafi? Okay, any further questions? So if there are no further questions, we will close this FDP here, and again tomorrow at 7 p.m. we will see you all. I think the feedback forms are already shared for the day 1 and day 2. You can fill it up, and the college management will be looking into it. Okay then, thank you so much. Thank you, Professor, again, and have a good time ahead. So we will be ending it here. I think the host can end it. I will. Indu madam, there is one question. Sir, if you are working on a specific gene of interest for any studies, how to select for the particular sequence length in a whole genome? Okay. Whole genome, I did not mention it here. So in whole genome, it is but so when you are telling it as a whole genome, you should not look for the particular length. That will be my suggestion. Because whole genome is already, it is like a few GBs. So for a few GBs, if it is there, it is, and above all, you should, a study should signify in whole genome analysis what exactly you are trying to find it out. If you are trying to find out the variants, or you are going to some other, what exactly is your the conclusion from the whole genome, that you have to understand. And the particular length doesn't come to the question when it is a whole genome sequence. The whole genome sequence, mainly it makes difference when you are working with the FASTA sequence, but not with the FASTQ sequence. Okay, any further questions? And above all, the specific length when you are taking, I will rather suggest that if at all you are working on the domain region, then it is important to specify the sequence length, and rather on the specifying the sequence length, you have to find out where exactly the domain regions are there. Then only it is apt to find out the sequence length. Otherwise, it is having least significance when you are doing the alignment. Okay, any further questions? Any further questions? Okay, if there are no further questions, the host can close the meeting.